Welcome to Tomorrow Unveiled, your weekly briefing on the science that is actively reshaping the human experience.
Hey there. Yeah, today we are really diving deep. Our dedicated research team has pulled together some incredibly compelling, robust breakthroughs from just this past week in longevity science.
And the focus, it feels like it's really sharpening, doesn't it?
It absolutely is. The whole field, it's shifting. It's less about chasing some theoretical maximum lifespan way out in the future.
Right, the immortality headlines.
Exactly. And it's much more focused on something, well, frankly, more valuable and immediate. Extending functional life.
Functional life, that's the key phrase, isn't it? It's everything. We're talking health span.
Precisely. The years where you have your full vitality, you're mobile, you're active, and maybe most importantly, your mind is sharp. Cognitive function is huge.
That's the goal. That's the metric that actually matters day to day. And the material the team compiled for Tomorrow unveiled this week. Wow, it covers so much ground.
It really does. It spans genetic engineering, some really practical cognitive science stuff, and these incredibly powerful new AI tools.
Yeah, the AI angle is fascinating. It's not just measuring aging anymore, is it? It feels like it's changing how we even understand what aging is.
That's a great way to put it. It's forcing a rethink. So today we're planning to unpack this across, let's say, 3 major frontiers.
Okay, lay it out for us.
All right. First, we'll dig into some amazing new insights about the body's own defenses, cellular and genetic defenses. We're talking about taking cues from supercentenarians.
People living past 110, incredible.
Yeah, and also mastering how cells clean themselves up, cellular housekeeping.
Okay, defense and cleanup, got it. What's next?
Then we move into more advanced therapies.
Yeah.
We've got a really astonishing finding, non-pharmacological, this one for rejuvenating the aging brain.
Non-pharmacological, that always gets my attention.
Mine too. And then we'll look at the absolute cutting edge of precision cell engineering.
And the third frontier.
And finally, we have to talk about the new rulers of the field, you could say, the measurement tools, specifically AI-driven systems, these aging clocks that are just accelerating discovery at a pace we've never seen before.
Right, and forcing us, as you said, to maybe redefine old itself. Okay. Sounds like a packed agenda. Let's dive in. Okay, so starting with those intrinsic defenses, I want to jump right into something that connects, well, the absolute peak of human longevity with, sadly, the most devastating kind of accelerated aging. This Supercentenarian gene variant, LAV-BPIFB4.
Yeah, this one really stands out. The core idea, I mean, just think about it. Taking a biological signal that's enriched in people who live healthily past 110.
Super healthy, usually.
Right, and figuring out how to translate that into a potential therapy for, well, for everyone else, or at least those facing age-related decline. It's pretty profound.
And the way the research team tested it is key here, right? They didn't just use standard aging models.
No, and that's what makes the findings so robust. They focused on Hutchinson-Gilford-Progeria syndrome, HGPS.
That's the condition where children age incredibly rapidly. It's heartbreaking.
It is. It's caused by a buildup of a toxic protein, progerin. So it provides this extreme accelerated aging test case. What the team did was administer just a single dose of this longevity gene variant.
A single dose?
Yeah, a single administration. They tested it in specialized mouse models that mimic HGPS and also in human patient cells cultured in the lab.
One shot. That really speaks to the potential potency, doesn't it? So what did they see? What were the immediate measurable effects, especially on the heart, which is hit so hard in HGPS?
The cardiac results were, well, frankly, they were striking. This single gene dose significantly improved heart function, specifically a really crucial measure called diastolic function.
Okay, diastolic function. Quick refresher for listeners.
So systolic function, that's the heart pumping blood out. Diastolic function is the heart's ability to properly relax and fill back up with blood between beats.
Okay, relaxation and filling.
Exactly. And as we age, naturally, the heart muscle tends to stiffen. Poor diastolic function is often one of the first and most dangerous signs of age-related heart failure.
So it's not just about the power of the pump, it's about the heart's ability to reset properly for the next beat, like taking a good breath.
That's a perfect analogy. The gene therapy helped restore that elasticity, that ability to relax and fill efficiently. But it did more.
Oh.
Yeah, it also dramatically reduced tissue scarring, what we call cardiac fibrosis, which is a major problem in aging hearts, and it actually promoted the growth of new, healthy blood vessels within that damaged heart tissue.
Angiogenesis. Wow. So it was basically reversing some pretty severe signs of age-related damage in the heart's structure and its performance.
In these specific models, yes, it was a remarkable reversal.
Okay, so that's amazing in itself, helping the heart in this extreme aging scenario. But here's the insight that our research team really highlighted, the thing that kind of changes the game therapeutically.
Right, it's how it achieved this.
Exactly. This gene variant, it didn't work by directly blocking or getting rid of that toxic progerin protein, did it?
No, it didn't. And that's what's so fascinating. If this were, a standard drug, you'd expect it to bind to progerin, maybe neutralize it, help clear it out.
The usual target and destroy approach.
Right. But instead, this gene seems to work by boosting the cell's own built-in resilience. It's intrinsic stress tolerance.
Intrinsic stress tolerance. So it makes the cell tougher.
Pretty much. It's almost like installing an upgraded operating system for the cell's basic functions.
Right.
This stronger OS lets the cell handle the damage better, whether that damage is from progerin or oxidative stress or chronic inflammation.
All the usual suspects in normal aging, too.
All the hallmarks of normal aging. So the cell becomes better equipped to tolerate the stress that comes its way.
Okay, hold on. If that's true, the implication is potentially huge, isn't it? Instead of needing, like, a different drug for every single bad molecule that builds up as we age,
You just make the cells fundamentally more robust.
so they can handle the inevitable messiness of aging better on their own.
That's the potential paradigm shift, exactly. You're moving from a reactive strategy, trying to block specific damage after it happens, to a more proactive one, enhancing the cell's inherent resilience before things get too bad.
Which could open up a whole new therapeutic avenue for things like heart failure, maybe other organ decline, that's focused on strengthening the cell's core defenses against a whole range of age-related insults, not just one specific toxin.
Precisely. It's a different philosophy of intervention.
And thinking about internal maintenance, that flows perfectly into the next finding about the cell's main waste disposal system, the lysosome.
Ah, yes, the lysosome, crucial little organelle.
We know generally that as we age, lysosomal function tends to decline. The cell's cleanup crew gets slow, less efficient.
That's right. And you get this buildup of, cellular garbage, molecular junk. This breakdown in proteostasis, the balance of proteins, is a key feature of aging.
Proteostasis, that's basically cellular housekeeping, right? Keeping things tidy.
Exactly. And when that housekeeping fails, junk accumulates. Things like lipofusin-age pigment and, of course, in the context of HTPS, that toxic progerin protein again. This buildup is a major driver of cells becoming senescent, those zombie cells we hear about.
Right. So the research team looked at this specifically in HGPS cells again.
They did. They identified a new therapeutic strategy focused on fixing the core problems with lysosomes seen in those patient cells.
So if the lysosomes are the recycling plants, and they're getting old and inefficient,
Cool.
how did the researchers jumpstart the cleanup? Did they just, force the existing plants to work overtime?
They went a step further, actually. They didn't just try to boost the existing lysosomes. They stimulated something called lysosome biogenesis.
Biogenesis, meaning making new ones.
Exactly. It's the cellular equivalent of building a whole new state-of-the-art fleet of recycling centers. They literally triggered the cells to create brand new, fully functional lysosomes.
Wow.
Yeah.
Okay. How?
And these new lysosomes then got to work clearing out the accumulated toxic junk, including progerin. They managed to achieve this stimulation using two quite different molecular methods, which is great because it helps cross-corroborate the mechanism.
Okay. What were the two methods?
So first, they stimulated a family of enzymes called protein kinase C, or PKC. PKC is involved in all sorts of signaling within the cell, and activating it, in this case, kicked off the machinery needed to build new lysosomes.
Okay. Stimulating PKC. And the second approach?
The second approach involved inhibiting a pathway that's, pretty famous in longevity circles, mTORC1, right? The mammalian target of rapamycin complex 1. That's a huge player. It senses nutrients, growth signals.
Is a master regulator. So why would inhibiting it, which usually slows down growth signals, help clear out toxic proteins? That seems counterintuitive at first.
It does, but it makes sense when you think about what mTORC1 does. When it's highly active signaling, hey, lots of nutrients, lots of energy, let's build stuff. It promotes cell growth and making new proteins, but it actually suppresses autophagy.
Autophagy, the cell's self-eating, the recycling process.
Exactly. So by temporarily inhibiting mTORC1, you flip that switch. The cell goes into a kind of stress response, conserve resources mode. It thinks, okay, maybe nutrients are low. Let's break down older damaged parts, recycle the components, clean house to survive.
Ah, I see. So inhibiting mTORC1 basically triggers this deep cleaning cycle.
Precisely. And in the study, doing this successfully triggered that cleanup, cleared out the toxic progerin and reduced the signs of senescence in the HGPS cells. It really reinforces how central this mTOR pathway is to the cell's ability to maintain its own health and integrity.
And it helps validate drug development strategies that are already focused on inhibiting mTORC1, like rapamycin or related drugs.
It's a powerful piece of validation, yes.
Yeah.
It suggests this is a really promising way to tackle the buildup of molecular junk associated not just with accelerated aging like HGPS, but probably with normal aging too.
Okay, moving up a level now, from inside the cell to the whole system, specifically blood and the immune system. The idea of young blood or factors circulating in the blood that influence aging. It's been around a while, sometimes a bit controversially.
It has. But our sources confirm some really significant findings regarding a specific protein, Platelet Factor 4 or PF4.
PF4, okay, what's its role?
Well, it's a protein that naturally tends to decline as we get older. And the research team showed it seems to be absolutely critical for properly regulating our blood-forming stem cells.
The hematopoietic stem cells, HSCs, the ones in the bone marrow that create all our blood and immune cells?
Those are the ones, and PF4 seems crucial for keeping them behaving correctly and also for reversing some aspects of age-related immune decline. You can almost think of PF4 as like the conductor of the orchestra for your blood and immune systems.
Okay, interesting analogy. So what happens in older individuals when the conductor kind of steps off the podium because PF4 levels drop?
Chaos. Well, maybe not chaos, but definitely dysfunction. The research showed that when PF4 levels go down, you lose that tight regulation. The aged hematopoietic stem cells start to lose their self-control. They begin to proliferate, to divide, kind of unchecked, and they become dysfunctional.
Proliferating unchecked. That doesn't sound good.
No, and it leads directly to a couple of major problems we see in aging. First, you get imbalanced immune cell production. You start making too many of certain types of inflammatory cells and not enough of the balanced adaptive immune cells you need for targeted responses.
Cued production.
Right. And second, related to that, you see a rise in systemic chronic inflammation, what people often call inflammaging.
Inflammaging.
Yeah.
That chronic low-grade inflammation that underlies so many age-related diseases. So the loss of PF4 contributes to that.
It appears to be a significant contributor, yes. It helps explain why the immune system often gets less effective and more inflammatory with age. It's not just wearing out, it's becoming actively unbalanced and, in some ways, overactive in the wrong directions.
Okay, but the exciting part is the intervention, right? What happened when they put PF4 back?
That's the really cool part.
Yeah.
When the team reintroduced PF4 to aged stem cells, and importantly, they tested this in both mouse and human stem cells, those old cells started acting young again.
Really? How so?
They regained their youthful, balanced function, they calmed down, stopped dividing so wildly, and started producing a healthier, more balanced mix of immune cells.
Wow. So the implication is if you could somehow restore PF4 levels or maybe target the pathway it uses, you could potentially delay that age-related immune decline. Make the whole immune system more resilient, more balanced.
That's the therapeutic hope, yes.
Yeah.
It suggests PF4 or its signaling pathway could be a really valuable target for tackling immune senescence. It wouldn't just be patching a problem. It might be restoring the system's original operating instructions, so to speak.
All right, so segment 1 gave us hope for the body's fundamental hardware. The heart getting more resilient, the cleanup crews getting boosted, the immune system getting rebalanced. But what about, the control center, the brain?
Ah, yes. Cognitive decline. It's arguably the thing people fear most about aging, the biggest threat to functional life, to health span.
Absolutely. Which makes one of the findings our research team flagged this week particularly uplifting, because it involves a measurable positive change in the brain achieved through a non-pharmacological intervention.
Yeah, this one is genuinely exciting because it points towards something actionable, accessible, and backed by hard physiological evidence. It comes from a clinical trial looking at older adults.
What did they have them do?
They had them engage in specific computerized brain training exercises. And we're not just talking about, doing crossword puzzles or Sudoku. These were highly targeted digital routines designed to challenge specific cognitive functions.
Okay, targeted training. For how long?
Just 10 weeks. But the crucial part wasn't just whether their test scores improved, though they likely did. The researchers actually measured a quantifiable change inside the brain in its chemistry.
How did they measure that? Brain scans.
Exactly. They used PT imaging, positron emission tomography, which is really the gold standard for looking at metabolic activity and neurotransmitter levels in the living brain.
And what did the PET scans show after 10 weeks of brain training?
They revealed a 2.3% increase in acetylcholine levels, specifically in brain regions critical for memory and attention.
Okay, 2.3% increase in acetylcholine. Now put that number in context for us. How significant is that?
It's actually incredibly significant when you compare it to the natural course of aging. Acetylcholine is a vital neurotransmitter, essential for focus, learning, memory consolidation, and the estimated natural age-related decline in acetylcholine in the human brain is roughly 2.5% per decade.
Wait, say that again. The natural decline is about 2.5% per decade.
Per decade, yes, that's the estimate.
Let me get this straight. This targeted non-drug digital exercise program lasting just 10 weeks, it effectively offset or reversed nearly an entire decade of average age-related decline in this absolutely critical brain chemical.
That is the staggering implication, yes. A 2.3% boost from 10 weeks of training almost perfectly matches the estimated 2.5% loss over 10 years.
Wow, that kind of reframes things, doesn't it?
It absolutely does. It tells us the brain retains significant neuroplasticity, the ability to change and adapt even in older age. And it shows that youthful brain chemistry, at least in terms of acetylcholine, can be measurably restored simply by providing the right kind of mental workout.
The right kind of targeted stimulus.
Exactly, the right exercise for the brain. And importantly, this is achieved without traditional drugs, which often just work by trying to prevent the breakdown of existing acetylcholine and can come with side effects. This suggests you can actually boost the supply.
It really challenges the idea that cognitive decline is just this inevitable one-way slide. It suggests the circuitry can be maybe not completely rebuilt, but certainly re-energized and resupplied. That's genuinely helpful.
It is. It encourages active intervention using the brain's own capacity for change.
Okay, so from digital training for the brain, let's pivot now to the absolute frontier of physical modification at the cellular level, precision engineering. The research team flagged a major technological leap here, a new system for modifying T cells called CRISPR-OF-CRISPRON.
Yes, this is a potentially transformative development, especially for the field of immunotherapy, using the immune system, specifically T cells, to fight cancer or autoimmune diseases.
We hear a lot about CRISPR gene editing, usually CRISPR-Cas9. How is this different?
So traditional gene editing, like the standard CRISPR-Cas9 system, works by creating a physical cut in the DNA strand at a specific location. You cut the DNA, and then the cell's repair mechanisms hopefully fix it in the way you want, maybe inserting new code or disabling a gene.
Revolutionary technology, but it comes with risks, right?
It does. Cutting DNA, especially in multiple places at once, carries inherent risks. There could be off-target cuts, toxicity, potentially causing chromosomal damage, or even triggering cancer itself. It's particularly challenging if you need to modify several genes simultaneously.
Which you often do need to do, right? Especially if you're trying to create really sophisticated immune cells, like armored T cells that can survive and fight effectively inside a solid tumor for a long time.
Exactly. You might need to turn off genes that cause exhaustion, turn on genes that enhance killing power, add genes for targeting. It gets complex fast. And trying to make multiple DNA cuts safely is extremely difficult.
So how does CRISPR-CRISPRON get around that cutting problem?
It avoids cutting the DNA altogether. Instead, it uses epigenetic editing.
Epigenetic editing. Okay, unpack that for us simply. We know DNA is the hardware code, the sequence. Epigenetics is more like the software, right? Instructions on how to read the DNA code.
That's a great way to think about it. The DNA sequence is the instruction manual for the cell. Epigenetic editing doesn't rewrite the words in the manual. Instead, it adds little sticky notes or highlights. Specifically, the system uses a chemical process called methylation.
Methylation adding small chemical tags to the DNA.
Right. These methylation tags act like signals. They can tell the cell's machinery, okay, ignore this gene chapter, effectively turning it off. That's the CRISPR-off part. Or they can remove tags to say, read this chapter louder, turning a gene on, the CRISPR-on part.
So you're controlling gene activity without actually breaking the DNA strand itself. Why is that inherently safer?
Because cutting the DNA forces the cell to use its complex and sometimes error-prone double-strand break repair machinery. That's where things can go wrong. Methylation, on the other hand, is a natural process the cell uses all the time to regulate genes. It's generally much less disruptive and more reversible.
And the researchers showed this works well.
Extremely well. They demonstrated they could safely and efficiently turn up to 5 different genes on or off simultaneously in human T cells. And they did this while maintaining high cell viability and stability, something that would be incredibly difficult, if not impossible, using DNA cutting methods without causing significant toxicity or chromosomal abnormalities.
5 genes at once, safely. That's a big leap in multiplexing capability. And do the changes last? Are the T cells functionally better?
Yes, the epigenetic changes were shown to be remarkably stable and durable. The cells effectively remembered the methylation patterns, even after dividing many times. And functionally, absolutely. For instance, they used CRISPR-OF to silence a gene called RASE2.
RASE2, what does that do?
It acts as a kind of molecular brake on T cell activation. T cells need brakes to prevent overreaction. But in cancer therapy, sometimes you want to release the brakes a bit.
Ah, so silencing RASE2 makes the T cells more persistent.
Exactly. By silencing that brake epigenetically, they created armored T cells that didn't exhaust themselves as quickly and maintain their ability to kill cancer cells for much longer.
Wow, being able to fine-tune the immune response like that, dialing activity up or down for multiple genes without risking the DNA itself, that really feels like the next generation of cell therapy. Safer, more potent, more durable.
It definitely points towards that future. It's a level of precision, cellular software engineering, not just hardware editing. It's breathtaking, really, and makes these advanced therapies feel much closer to reality.
It absolutely does. Okay, before we move on to the crucial topic of measurement and how AI is changing everything, let's just take a quick pause. If you're finding this insightful, maybe learning something new, please take just a moment to like this video, subscribe to Tomorrow Unveiled, and maybe even consider hitting that new hype button. Apparently it helps us reach more viewers. And definitely turn on notifications so you don't miss our future updates.
And if you're listening to Tomorrow Unveiled on a podcast service, perhaps Apple Podcasts or Spotify, we would be incredibly grateful if you could leave us a rating and maybe even a short review. It genuinely helps new listeners discover the show.
Okay, we've talked about some really exciting potential interventions, from gene variants to brain training to epigenetic editing. But the big question always is, how do we know if these things are actually working in a meaningful way for people?
Right. You need rigorous measurement. And the technology for measuring aging itself is evolving incredibly rapidly. It's really professionalizing the whole field.
So let's start with a classic biomarker, telomeres, but link it to a very common, accessible intervention, vitamin D.
Okay. This finding comes from a large, well-regarded clinical trial called the VITAL study, specifically a sub-study focused on aging markers. They looked at the long-term effects of daily vitamin D3 supplementation.
How much vitamin D?
It was 2,000 IU per day, taken over 4 years. And the research team tracked nearly 1,000 older adults, specifically looking at what happened to the telomeres in their white blood cells, their leukocytes.
Telomeres, those protective caps on the ends of our chromosomes, they get shorter each time a cell divides, kind of like the plastic tips on shoelaces fraying over time.
Exactly. Shorter telomeres are generally considered a marker of cellular aging. So did the vitamin D help?
That's the question. Was the result positive?
It was, yes. The group taking vitamin D showed significantly less telomere shortening, less attrition, compared to the group taking a placebo over those 4 years.
How much less? Was it a meaningful difference?
It was statistically significant and arguably biologically meaningful, too. They preserved about 140 extra base pairs of telomere length compared to placebo. Now, putting that in context.
Okay, what does 140 base pairs mean?
Well, estimates suggest telomeres naturally shorten by maybe around 460 base pairs over a typical decade. So preserving 140 base pairs over 4 years, it's roughly equivalent to delaying that specific aspect of cellular aging by nearly 3 years.
Wow, okay. Almost 3 years of delayed cellular aging measured by telomeres just from taking a standard dose of vitamin D.
That's what the telomere data suggests, and the likely mechanism, probably related to vitamin D's known anti-inflammatory effects, which could help stabilize DNA and protect those telomere ends.
So on the surface, that sounds fantastic, right? An inexpensive, readily available supplement slows down a key marker of cellular aging. Seems like a clear win.
It does seem like that.
Yeah.
But, and this is a really crucial but that the research team strongly emphasized, we'd run smack into the biomarker versus outcome dilemma.
Ah, the gap between what happens at the cellular level and what happens to the whole person's health. Explain that dilemma here.
Okay, so while this sub-study showed a nice improvement in the biomarker, the telomere length, the main parent VITAL trial, which was much larger and designed to look at major health events, well, it did not find a statistically significant reduction in the things people really care about.
Like what?
Like preventing major cardiovascular events, heart attacks, strokes, or reducing the risk of getting diagnosed with cancer. The vitamin D didn't seem to move the needle significantly on those hard clinical outcomes.
Huh. Okay, so my telomeres might look 3 years younger on paper after taking vitamin D, but my actual risk of having a heart attack or getting cancer didn't really change in that time frame.
According to the main trial results, correct.
So, what good is the biomarker then? Why should we even care about the telomere result if it doesn't translate into better health outcomes, at least in this study?
That is exactly the right, critical question to ask, and it highlights why we have to be so careful interpreting biomarker data in aging research. It's a huge challenge for the field.
Why might there be a disconnect?
Several possibilities. Maybe 4 years is long enough to see a change in a relatively fast-moving cellular marker like telomere length, but it's just not long enough to see a difference in major diseases that develop over decades.
Okay, timescale matters.
Definitely. Or maybe telomere length, while correlated with aging and health, isn't actually the direct cause of those specific diseases. Perhaps vitamin D's anti-inflammatory effect helps telomeres, but maybe it wasn't strong enough or targeted enough to overcome all the other factors driving heart disease or cancer risk in that population over 4 years.
Right, it's complex. Inflammation affects both, but maybe fixing the telomere part isn't enough if other inflammatory drivers remain.
Precisely. It underscores the need for smarter, more comprehensive ways to measure aging and the effects of interventions. Which brings us very nicely to the AI revolution.
Yes, the tools that can handle this complexity. Our sources highlighted a specific AI platform, the Cadence Analyst, being used in collaboration with university researchers. And it's doing more than just crunching numbers faster.
Much more. This platform isn't just analyzing existing data sets in, days instead of years, which is already amazing. It actually generated a fundamentally new hypothesis about the nature of aging itself. One that really challenges the way we've thought about it for decades.
Okay, what's the hypothesis?
It's the concept of staged biological program.
Staged aging, like progressing through different phases. How does that differ from our usual view?
Well, we typically think of aging as this sort of continuous linear decline, right? A slow, steady accumulation of damage, things gradually breaking down over time.
Yeah, a gradual slope downhill.
The AI analysis suggests that model might be wrong, or at least incomplete. It indicates that aging might actually progress through distinct, definable biological stages or phases, kind of like how human development progresses through clear stages: infancy, childhood, adolescence, adulthood.
Huh. So different biological processes might dominate at different points in the aging trajectory.
That's the idea. The AI found that the sets of genes whose activity patterns are most predictive of your biological age actually change depending on where you are in your lifespan.
Give me an example.
Okay, maybe in your 30s or 40s, the most important predictors of your biological age might be related to metabolic pathways or growth hormone signaling. But then once you get into your, say, mid-60s or 70s, a completely different set of genes might become dominant predictors. Maybe genes related to clearing out senescent cells, or regulating inflammation, or managing immune function.
Whoa. Okay, if that's true, if the key drivers of my biological age in my 30s are different from the key drivers in my 60s, the implications for anti-aging strategies are massive.
Massive is right, because it strongly suggests that a one-size-fits-all anti-aging drug or intervention taken consistently throughout life is probably doomed to fail. Or, at best, it might only offer marginal benefits during one specific stage where it happens to target the relevant pathway.
So you can't just take, say, metformin from age 30 to 90 and expect it to be optimal effective the whole time.
Probably not, according to this hypothesis. The intervention needs to be timed precisely according to the individual's specific biological age stage. You might need something targeting metabolism earlier in life, and then something completely different, maybe targeting senescence or immunity later on.
Which means we absolutely need those highly precise AI-informed diagnostic tools to tell us which stage we're in.
Exactly. You need to know which biological program is currently running the show in your body to intervene effectively.
And those kinds of diagnostic tools, the aging clocks, are getting incredibly sophisticated. Let's talk about the spectrum, maybe starting with the high-tech MRI clocks and then moving to the more accessible wearable clocks.
Great. Yeah, the MRI-based aging clocks are really impressive demonstrations of AI power. These models are trained on thousands of high-resolution MRI scans of multiple vital organs: brain, heart, liver, kidneys, lungs, etc.
So not just one overall biological age number?
No, that's the key difference. They generate organ-specific age gaps. So the AI might look at your scans and say, okay, chronologically, you're 55, But your liver MRI looks like that of an average 62-year-old, so your liver age gap is plus 7 years. However, your brain scan looks more like a 53-year-old's, so your brain age gap is net as 2 years.
That level of granularity, that seems incredibly useful.
It is, because those organ-specific age gaps are proving to be very accurate predictors of future health outcomes. If your liver consistently shows an older biological age, that points to a specific vulnerability. It can help link systemic risks, like maybe chronic inflammation or hypertension, directly to the organ that's being most affected.
So doctors could potentially use this to say, okay, your biggest issue right now seems to be accelerated aging in your kidneys. Let's focus our preventative efforts there.
Exactly. It moves us closer to truly personalized preventative medicine by identifying the weakest link in the system much earlier.
Okay, but like you said, most of us aren't getting full-body MRIs every year. That's still pretty high-end. What about tools for everyday tracking?
And that's where the wearable-based aging clocks come in, and where we're seeing the real potential for democratization of this kind of monitoring.
Using data from fitness trackers, smartwatches.
Primarily, yes. Algorithms are being developed that use data readily available from common consumer wearables. A key input seems to be cardiovascular data, especially metrics related to vascular stiffness, which can often be estimated using the PPG sensor, the little flashing light on the back of your watch that measures blood flow.
PPG, photoplethysmography.
Yeah.
Right, so changes in blood vessel stiffness reflect aging.
They do. Arteries tend to stiffen with age. So algorithms like one called PPAge can analyze patterns in your PPG signal over time to estimate your vascular age, which correlates surprisingly well with overall biological and chronological age.
How accurate are these wearable clocks? And how sensitive can they pick up on shorter-term changes?
They're getting remarkably accurate at predicting chronological age, often within a few years. And yes, they seem quite sensitive. The research team noted examples where these clocks detected significant acute biological shifts.
Like what?
Things like the temporary but measurable increase in biological age sometimes observed during pregnancy, or following a major physiological stressor like surgery or even a severe infection.
So they can pick up on those big events, but can they also show improvements?
Yes, and that's the really powerful part for motivating lifestyle changes. Studies are showing that people who make sustained improvements may be consistently hitting exercise goals, improving sleep quality, managing stress, maybe eating better, can actually see their calculated biological age gap decrease.
Really? By how much?
Reductions of maybe 3 to 4 years compared to their chronological age aren't uncommon in people making significant positive changes over time.
Okay, that's huge for feedback. Seeing your age go down on your watch because you stuck to your running schedule. That's a powerful motivator.
It really is. It democratizes the monitoring, puts actionable data directly into the hands of the individual, and provides that immediate feedback loop that can be so crucial for sticking with healthy habits.
Okay, so we've covered this amazing range of potential breakthroughs. We've got accessible things like maybe vitamin D having some effect or targeted brain training.
Right.
All the way up to incredibly complex high-tech stuff like the LAV-BPIFB4 gene therapy or the epigenetic T-cell editing.
A huge spectrum.
Which brings us inevitably to the really thorny but maybe most important question. If we actually succeed in significantly extending functional life, who gets to benefit?
Yeah, the access question, it looms large over the entire field, and the analysis from our research team really highlights this growing divergence, this potential chasm in the longevity ecosystem.
A chasm based on cost and complexity.
Exactly. On one side, you have potentially low cost, relatively safe, widely accessible interventions, maybe like the vitamin D findings or lifestyle changes guided by wearable data, things most people could potentially access.
Okay, the democratic end of the spectrum.
Right. But on the other extreme, you have these cutting-edge interventions we discussed, the gene therapies, the advanced cellular engineering. These require immense research, development, manufacturing infrastructure. The projected costs, at least initially, could be astronomical.
We're talking hundreds of thousands, maybe even millions of dollars or euros per treatment.
Easily, based on current costs for similar advanced therapies. And this huge cost difference is already fueling something else.
The boom in private longevity clinics.
Precisely. We're seeing this explosion of private clinics popping up globally. They often cater to wealthy clients, offering these bespoke, sometimes very aggressive anti-aging regimens.
What kinds of things are they offering?
It can be a huge range, maybe complex cocktails of peptides, massive doses of various supplements, often without strong evidence, sometimes even unproven stem cell therapies. And the price tags can be staggering. You hear figures from, say, 10,000 to well over 100,000 per year.
Wow, and the scientific validation for many of these clinic protocols is questionable.
Often, yes. Many operate in a kind of regulatory gray area. There's limited, independent, peer-reviewed evidence for the specific combinations or dosages they're using. So you have serious concerns about not just efficacy, but also safety and, of course, equity.
Which brings us back to the core ethical dilemma. If the interventions that truly offer the potential for, say, an extra decade or two of healthy, functional life, maybe those high-tech gene or cell therapies down the line, if those are only accessible to the people who can afford that 100,000 plus price tag, what happens to society?
You create a biological divide. The gap between the longevity haves and have-nots wouldn't just be about wealth anymore. It would be about fundamental differences in health span, in functional years lived. That's a recipe for potentially massive social inequality and instability.
A chasm of biological inequality. That's a sobering thought.
It is. And access is often tightly linked to risk profile, right? The riskier an intervention, generally the more restricted its initial access.
Okay, so let's compare the risk profiles we've discussed. Vitamin D.
Generally very low risk at standard doses like 2,000 IU.
Yeah.
Widely available, low cost. The main risk there is perhaps overstating the benefits based only on biomarker data, like the telomere finding, before we have solid proof of clinical outcomes. Responsible communication is key.
Okay. Now what about the other end? The epigenetic T-cell editing using CRISPR-OF-CRISPRON.
That falls squarely into the high cost, high complexity, and still inherently high risk category.
Yeah, you said it was safer because it doesn't cut the DNA.
It is significantly safer than DNA cutting methods in terms of genomic damage. That's a huge advance. But it's still a very advanced cellular therapy. You're taking a patient's cells out, genetically modifying them, even epigenetically, growing them up and infusing potentially billions of them back into the body, often using viral vectors for delivery.
And that process carries risks regardless of the editing method.
Absolutely. There are risks associated with the cell manufacturing, the delivery vectors themselves, and even with epigenetically modified cells, if you're creating, say, supercharged immune cells, there's still the risk of the immune system overreacting. Things like cytokine release syndrome, CRS, or potential neurotoxicity, these are known risks with powerful T-cell therapies, regardless of how the T-cells were engineered.
So because of those inherent risks, these powerful tools won't be used for general anti-aging anytime soon.
No way. They'll be restricted, quite rightly, to treating severe, often life-threatening diseases first. Things like advanced cancers where the potential benefit clearly outweighs the significant risks.
And only after years of proving safety and efficacy in those really sick patient populations could they even begin to be considered for broader, maybe preventative or health span extending applications in healthier people.
Exactly. The regulatory bar for using these kinds of potent therapies to add healthy years to someone who isn't already facing a catastrophic illness is and should be incredibly high. It necessitates a very cautious, staged rollout over many, many years.
Okay, so we've got this landscape.
Yeah.
Some accessible, likely incremental gains available now or soon. Then these potentially transformative but high risk, high cost, and initially very exclusive interventions further down the road. How does the research team see this converging? What's the likely path forward in the next, say, 3 to 10 years?
The strong consensus emerging from the research we reviewed points towards the absolute necessity of combination therapies.
Meaning hitting multiple aging pathways at once.
Precisely. It seems increasingly unlikely that there will be a single magic bullet drug or intervention that targets just one mechanism and dramatically slows aging across the board. The process is just too complex, too multifaceted.
And we're already seeing evidence for this in animal studies.
Yes, there's strong preclinical evidence. Studies combining, for example, rapamycin, that MTR inhibitor we talked about, with other compounds targeting different pathways often show additive or sometimes even synergistic effects. The combined benefit is much greater than either drug alone.
So the future of effective functional life extension probably isn't a single pill, but more like a carefully personalized cocktail of interventions.
A carefully orchestrated cocktail, yes. And crucially, as the staged aging hypothesis suggests, a precisely timed cocktail.
Bringing the timing aspect back in.
Absolutely. So, looking ahead, we anticipate seeing the first major clinical trials for some of these advanced gene therapies maybe LAV, BPI-FB4 for heart issues, perhaps clotho-protein analogs launching within the next, say, 1 to 5 years. But they'll initially target specific severe diseases like heart failure or maybe even HGPS to gather that vital safety and efficacy data.
And underpinning all of this, enabling the combination approach and the timing, will be the AI platforms.
They become essential infrastructure. Tools like the Cadence Analyst or similar AI systems will be critical for driving the adoption of that staged aging model. They'll be needed to analyze an individual's multi-omic data, genetics, epigenetics, proteins, metabolites, along with maybe imaging and wearable data to determine their current biological age stage.
And then recommend the right combination of interventions for that specific stage.
That's the ultimate goal. For example, the AI might determine that for you, right now, pathways related to immune function and inflammation are the dominant drivers of your aging. So maybe an intervention related to PF4, perhaps combined with something to clear senescent cells, would be the most effective strategy at this point in time. But 5 years earlier or 10 years later, the optimal combination might be completely different. Hashtag tag outro. So just to wrap up the key takeaways from this week's deep dive on Tomorrow Unveiled. First, and maybe most importantly, there's this fundamental shift happening. The focus in longevity science is now squarely on functional life extension, maximizing health span, the quality of years, not just chasing the maximum number.
Yeah, quality over just quantity. Second, we saw breakthroughs happening across multiple, really complementary fronts. There's hope for boosting the body's resilience, like with the LAV, BPI, FB4 gene for cardiac health, or rebalancing the immune system with PF4.
Right. And then there's the cognitive side, that amazing finding about reversing nearly a decade's worth of acetylcholine decline with just 10 weeks of targeted digital brain training. That's huge.
Measurable, non-drug improvement. And
Third, measurement is king, and it's getting incredibly sophisticated. AI-driven aging clocks, validated biomarkers like telomeres, they're professionalizing the field rapidly.
All right.
But as the vitamin D study showed, we have to stay critical. We need to demand that promising biomarker results eventually translate into real-world, confirmed clinical outcomes. We can't just chase markers.
Absolutely. Healthy skepticism is crucial. Okay. As always, we love hearing what you think. If you're watching on YouTube or listening on your favorite podcast app, please drop a comment. Let us know your thoughts on the future of longevity. How do we navigate these breakthroughs? How do we ensure they become accessible to everyone?
Yeah, we really value that input.
And here's a final thought to leave you with, building on that AI discovery. If the research team's finding really holds up this idea that aging isn't linear, but is actually a staged biological program, what does that mean for how you should be thinking about managing your own health right now and in the future?
That's a big question.
Do we need to fundamentally move beyond the generic lifelong advice, eat well, exercise, sleep well, and basically wait until these precision aging diagnostics are available to truly optimize our vitality at each specific phase of life? Or is the best strategy for now still just doubling down on mastering those basics, building the strongest foundation possible while the more advanced personalized technology catches up? Where's the balance? That's something we'll definitely keep exploring here on Tomorrow Unveiled.